Hello guys, welcome to my channel. So as you know, we are still studying about the protein structure and function. If you saw the previous video, we studied about the different methods, sorry, that have been used to detect the amino acid sequence in the peptide chain or a protein. So now we will study about the three-dimensional structure of protein. All right. So proteins are big molecules. So the covalent backbone of a typical protein contains hundred of individual bonds, right? And because free rotation is possible around many of these bonds, the protein can, or in principle, assume a virtually uncountable number of conformations. So to understand the structure of protein, there are six themes actually. So the first is that the three-dimensional structure or the structures taken up by a protein are determined by its amino acid sequence. Second, the function of typical protein depends on its structure. Third, the most isolated protein exists in a one or a small number of stable structural forms. Fourth one is the most important forces stabilizing the specific structure maintained by a given protein are non-covalent, and the hydrophobic effect is particularly important as well. Fifth is amid the huge number of unique protein structure, we can recognize some common structural patterns that help to organize our understanding of protein architecture. And the sixth final one is the protein structure are not static. All right, so to understand the structure of protein, so the main thing is the spatial arrangement of atoms. So I'll write it down. Sure. Arrangement of atoms, or it can be the arrangement of atoms in a arrangement of atom in a protein that is in a space, or any part of a protein. Any part of a protein is called its conformation. So that is called a conformation. So any arrangement of atom in space of a protein is called its conformation. So the possible conformation of protein or protein segment include any structural state it can achieve without breaking the covalent bond. So what are the possible conformation or structural or a space arrangement of atoms of in a protein are that includes structural state it can achieve without breaking any covalent bonds. So the need for multiple stable conformations reflects the changes that must take place in the most protein as they bind to other molecules or catalyzes the reactions. So the conformations existing under a given set of conditions are usually the ones that are thermodynamically most stable. That means that have the lowest Gibbs free energy. So proteins in any of their functional folded conformation are called native protein. So what are native protein? So protein in any of their functional folded conformation are called native protein. Native proteins. All right, so these are known as the native protein. So that means proteins in any of their functional folded conform. So there will be a different conformations for different functions. All right, so in the protein structure, the term stability can be defined as tendency to maintain a native conformation. What does that stability means in context of the protein structure? That means the tendency to maintain a native conformation. So that means native proteins are only marginally stable. Why? Because the delta G, so that's the free energy, delta G, that's the free energy for separating the three-dimensional protein and the unfolded states in a typical protein under the physiological condition is in the range of, what's the range of this, is 20 to 65 kilojoules per mole. So this is the range for separating the folded and unfolded state in under the physiological condition. But a given polypeptide chain can theoretically, just the theoretically, assume countless conformation, and as a result the unfolded state of a protein is characterized by the high degree of conformational entropy. So that means this entropy, along with the hydrogen bonding interaction of many groups in the polypeptide chain with the solvent, that is the water, tends to maintain the unfolded state. Right? So the chemical interaction that counteract these effects and stabilizes the native conformation, including, so what makes the native conformation stabilize? All right, so this things stabilizes the native conformation, that is disulfide bond, weak interaction, that are the non-covalent bonds, and the forces, that is hydrogen bonds, hydrophobic effect, and the ionic interaction. But many proteins do not have the disulfide bond. So if you guys don't know what is disulfide bond, so the disulfide bond is a S2S bond, that is the sulfur to sulfur bond. So I'll just show the example. So for example, that this is the sulfhydride to sulfhydryl group, and when it is oxidized, it will form this disulfide bond with each other, that will have two hydrogen and two electrons. Sulfhydryl group, these are the sulfhydryl group. So that is also known as thiol group. So that indicates that the sulfur is bonded to a hydrogen atom, right? And the R represents only the alkyl or other organic substituents. All right. So this was just for the example. Right, so we'll go back. So many proteins do not have the disulfide bonds, but the environment within the most cell is highly reducing due to high concentration of reductants. That means a reducing agent. So in the most cell, the environment is highly reducing. That means it has a high concentration of reductants such as glutathione, and that because of that reducing agent, the most sulfhydryls, this sulfhydryl will thus remain in the reduced state. It will not form this oxidized state. But outside the cell, the environment is more oxidizing, and the disulfide formation is more likely to occur. So in eukaryotes, disulfide bonds are found primarily in secreted extracellular protein. For example, the hormone insulin. And there you can see this disulfide bond. But the disulfide bonds are also uncommon in bacteria proteins as well. So why we studied this? Because for all proteins of all organisms, weak interactions are especially very important in folding of the polypeptide chain in their secondary and the tertiary structures. Right, so what happens, so when we saw the just a linear chain of a polypeptide and how it goes into the secondary and the tertiary structure, it's because of the weak interactions, the association of multiple polypeptides. So when there are multiple polypeptides, that will form a quaternary structure, and that also relies on the weak interaction. So weak interaction, so this secondary and the tertiary structure depend on the weak interactions, that is the non-covalent bonds. So about 200 to 460 kilojoules per mole are required to break a single covalent bond, right? So the energy to break the covalent bond is 200 to 460 kilojoules per mole. All right. So but for the weak interaction, the weak interaction can be broken by just merely the force of 0.4 to 30 kilojoules per mole. All right. So this is for weak interaction and this is for covalent bonds. All right. So this is the energy required to break the covalent bond and this is the energy required to break the weak interaction. Right. So individual covalent bonds such as the disulfide bond linking separate parts of the single polypeptide chain are clearly much stronger than the individual weak interaction. So now we understand why it's much stronger the disulfide bond that are linking in the polypeptide chain, because the breaking of these covalent bonds the energy is required really high, but when there are weak interaction for the polypeptide chain in between, the energy required is really low. Yet with the weak interaction, as in polypeptide chain there are so many weak interactions in the chain that the stabilizing force in protein structure is depends on the weak interaction. So this depends on the weak interactions. All right. And in general, the protein conformation with the lowest free energy, that is the most stable conformation, is one with the maximum number of weak interactions. All right, so the stability of protein is not simply that's just the sum of free energies of formations of many weak interaction within it. So, but on carefully examining the contribution of this weak interaction to protein stability, that we find that the hydrophobic effect generally predominates. We know that there are proteins that have hydrophobic amino acids, right? So that hydrophobic amino acids, how they behave. So for example, in pure water contains a network of hydrogen bonded H2O molecules. And if you remember, we studied for the lipids as well, for the hydrophobic and the hydrophilic fatty acid molecules. We studied that in the cell membrane. So when hydrophobic is going into the water, how the water will react, how it form a meshell around it. And so in the same way for this protein as well. So when the hydrophobic part of the protein is in the water, so the what the water will do, so when the water surrounds a hydrophobic molecule, the optimal arrangement of hydrogen bonds results in a highly structured cell. So like as we know, like when there is a hydrophobic, for example, this is a hydrophobic part or a hydrophobic protein, like, and it's in the water, so the water will form a hydrogen bonds around it. This layer or a shell is known as solvation layer. Solvation layer of water around the molecule. So the increased order of water molecule in the solvation layer correlates with an unfavorable decrease in the entropy of the water. So the randomness of the water decreases and the increased order of the water molecule around it increases. So however, but when the non-polar group clusters together, the extent of solvation layer decreases because each group no longer presents its entire surface to the solution, right? So the amino acid sequences of most protein thus include a significant content of hydrophobic amino acid side chain, like for example leucine and valine, tryptophan. So this have all the hydrophobic content of amino acids. So they do like this, and these are positioned so that they are clustered when the protein is folded, forming a hydrophobic protein core. So this is the one reason for folding the protein. But under the physiological condition, the formation of hydrogen bonds in a protein is driven largely by this same entropic effect with the same randomness effect. So polar groups can generally form, you know that the polar groups generally form hydrogen bonds with the water, right? When it goes into the water, the polar groups form the hydrogen bonds with the water and hence are soluble in water. They are known as hydrophilic, right? However, the number of hydrogen bonds per unit mass is generally greater for pure water than for any other liquid or solution. So the number of hydrogen bond is really greater in the pure water than any other liquid or solution. And there are limits, but still there are limits to the solubility of even the most polar molecule as their presence causes a net decrease in hydrogen bonding per unit mass. So it's when any polar molecule is introduced to the water, it's not that all the polar molecules will form a hydrogen bonds. So there is a limit to that as well. And this so the net solubility of even the most polar molecule as their presence causes a net decrease in hydrogen bonding per unit mass. Therefore, a solvation layer forms to some extent even around the polar molecules. So it's not all the polar molecule that goes into the water interact with the water and just form hydrogen bonds. But there are some polar molecules around that polar molecule, solvation layer still forms around that. So although the energy of formation of intramolecular hydrogen bond between two polar groups in a macromolecule is largely cancelled by the elimination of such interaction between this polar molecule and the water, the release of structured water as an intramolecular association forms provide an entropic driving force for folding. So that means the hydrophobic effect is clearly important in stabilizing the conformation. This arrangement and the interior of the structured protein is generally densely packed core of hydrophobic amino acids. So when there is a three-dimensional structure, the interior is the densely packed hydrophobic amino acid chains. And it is also important that any polar or charged groups in the protein interior have suitable partners for hydrogen bonding or ionic interaction. But in addition, the hydrogen bonds between groups in a protein form cooperatively, like that means formation of one make confirmation formation of the next one in the repeating secondary structure. So what happens in the hydrogen bonds between groups also in a protein forms cooperatively. So if there's one hydrogen bond forms, then as soon as the first form, then there will be a second hydrogen bond will form. And that will be for the secondary structure that optimizes the hydrogen bonding. So in addition, the hydrogen bonds between groups in a protein form cooperatively in repeating secondary structures that optimizes hydrogen bonding. So in this way, hydrogen bonds often have an important role in guiding the protein folding process. So the interaction of oppositely charged groups that form an ion pair or a salt bridge can have either a stabilizing or destabilizing effect on a protein structure. So the ionic interaction also limit the structural flexibility and confirms a uniqueness to a particular protein structure that the clustering of non-polar groups via the hydrophobic effect cannot provide, right? So in the tightly packed environment of the protein, there is one more type of weak interaction that can have a significant effect, and that interaction is known as van der Waals interaction. So do you know this interaction? So this interaction means a dipole to dipole interaction. For example, just a simple example, if this is one atom, so that will have a positive and negative charge, and this is the another atom that will have the same positive and negative charge. And if these two atoms are really close, the I think the distance would be like zero point 0.5 nanometers something, and so when they are this much close, it will develop an interaction with each other. So that dipole to dipole interaction is known as the van der Waals interaction, right? So the van der Waals interaction or dipole to dipole interaction involving the permanent electric dipoles in groups such as carbonyls. The transient dipole derived from the fluctuation of electron cloud surrounding an any atom, and dipole induced by interaction of one atom with another, that is a permanent or transient dipole. So that is the van der Waals interactions. So as the atom approaches each other, this dipole to dipole interaction provides an attractive intermolecular forces that operates only over a limited intermolecular distance, as I mentioned, and that is around zero point 0.3 to zero point 0.6 nanometer, 0.3 to 0.6 nanometer. So that's the distance. But the van der Waals interactions are really weak and individually contributes little to overall protein stability. So that do not very much qualify for the protein stability. However, in a well packed, but the when the protein is well packed, surrounded, or in an interaction between a protein and another protein or other molecule at a complementary surface, the number of such interactions can be substantial. So from the structural patterns, what it reflects that hydrophobic residues are largely buried in the protein interior, that is away from the water, and number of hydrogen bonds and the ionic interactions within the protein is maximized, thus reducing the number of hydrogen bonding and ionic groups that are not paired with a suitable partners. Alright guys, so we studied about the weak interaction, van der Waals interaction that are in the protein structure. So now there are covalent bond as well that we know, and that plays also an important constraint on the conformation of a polypeptide. So in late 1930s, who laid the foundation? So in 1930, the name is Linus Pauling and Robert Corey. I think it's that right, Robert Corey. Right, these two laid the foundation for the current understanding of protein structure. So what these guys found, so I will just draw a small diagram here, or rather a structure. So this is of a carbon atom, for example. This is oxygen, right? This is nitrogen, hydrogen, and it's something like this. I am not that perfect in diagrams, but it goes something like this. Then there will be like this again. It actually goes like this, this, and this. So this is like same thing like here, and so on. There will be, for example, I would one more here. C alpha. All right. So the Linus Pauling and Robert Corey, they began a careful analysis of the peptide bond, and they found that the alpha carbon atom of adjacent amino residue are separated by 3 covalent bonds. So they are arranged like this in a simple plane. So this is alpha carbon, then carbon, then nitrogen, and then again alpha carbon. Right, that you can see here, alpha carbon, carbon, nitrogen, and then alpha carbon. So they are the alpha carbon of 2 amino acid residues are separated by 3 covalent bonds, 1, 2, and 3 covalent bonds, right? So, and this shows in the X-ray diffraction studies as well, that of simple dipeptide and tripeptide showed that the peptide bond that is between this carbon and nitrogen is somewhat shorter than the carbon and nitrogen of a simple amine group. So if this is a peptide bond of carbon and nitrogen, but there are simple amine group as well that have carbon and nitrogen, but the peptide bond for this carbon and nitrogen are shorter than that of the amine group, and that the atoms associated with peptide bonds are coplanar. So the atoms associated with this peptide bonds, all of these are coplanar, means they are in just one same plane. So because of this coplanar, so that indicate a resonance or partial sharing of 2 pairs of electrons. So the 2 pairs of electrons between the carbonyl group and the amide nitrogen group. So this is the carbonyl group and this is the amide nitrogen group. So the oxygen, because of that, the oxygen has a partial negative charge, and the hydrogen bonded to the nitrogen has a net partial positive charge here, setting up a small electric dipole. So that's the electric dipole, means this positive and this negative will have some interaction or will create a small electric dipole, right? So the 6 atom of peptide, so this is a 6 atom, right? 1 2 3 4 5 6 atoms of the peptide group lie in a single plane, with oxygen atom of carbonyl group trans to the hydrogen atom of amide group. Right, so I will repeat it again. So that means the 6 atom of peptide bond lie in a single plane. So that's a single plane with oxygen atom of carbonyl group trans to the hydrogen atom of amide group. So what is meant by trans if you don't know this? So in organic chemistry, this term is used cis trans isomerism, or it is also called known as a geometric isomerism or configurational isomerism. So it's just simple. So the cis means in Latin the side of, and the trans means the other side of. So in chemistry it indicates that this functional group are on the same side of the carbon atom for cis. So for example, so just for your knowledge, right? So for example, I'm just drawing a random H3C, right? CH3. So this is an example of cis, right? So in this example, that means the functional group. So these are the functional group are on the same side of the carbon atom, or sorry, rather the carbon chain. So these are on the same side of the carbon chain, not on the opposite side. But for the trans, for this trans, this is the case. This is not the case. So for the trans, right? So we'll try it here. Trans. All right. So for the trans, it looks like this. So for example, this is the carbon chain. All right, and it will be on the opposite side or the other side. So the trans means functional group are on the other side of the carbon chain. So this is on this side and this is on the other side. So for the polypeptide bond or the peptide bond as well. So the carbonyl group is a trans to the hydrogen. So for it's the same case for the peptide group or the peptide bond rather. So the oxygen atom of a carbon or the rather carbonyl group trans to the hydrogen atom of the amide group. So they are on the other side, right? Okay, so I will just remove this. Right. Okay. So from this finding, Pauling and Corey concluded that the carbon and the nitrogen bond, because of their partial double bond character. Why they have double bond? Because this is one bond, right? Carbon to nitrogen. But as they have the partial negative and the partial positive charge, this is also a one more bond. So that's why it's called a partial double bonded character. Because of this character, they cannot rotate freely. So this part cannot rotate freely. So rotation is permitted. Where the rotation is permitted, it's permitted on this side, nitrogen to carbon alpha or carbon alpha to the carbon bonds, right? But this part rotation is not permitted. So the backbone of polypeptide chain can thus be pictured as a series of rigid plane. So that's why the peptide bond has a rigid plane with consecutive plane sharing a common rotation point at, so this is the rotation point here. That's a rotation point here at the carbon alpha. The rotation points are here. So this is the carbon alpha rotate, and this is the rotation points here. That's here, and then will be on the nitrogen and carbon rotation point. All right. Then again on this side, it's the same. So these are the rotation points. Right. So the peptide bond conformation is also defined by three dihedral angles. So the peptide bond conformation is also defined by three dihedral angles. So, and this dihedral angle, also known as dorsal angles, called by this symbol, that is the phi, then this symbol, that is the psi, and omega. Right, so how it is reflecting the rotation? So this reflect the rotation about each of the three repeating bonds in the peptide backbone. So first, what is the dihedral angle if you don't know? So the dihedral, angle is the angle between the two intersecting plane, or in chemistry is the angle between the two planes through the two sets of three atoms having two atoms in common. So we'll just see an example, a simple example. So just draw a picture here. So just for example, this is a 3D view of some chain. All right. So from this atom, all right, you can see, right? So from this atom to this, so that's a dihedral angle, and from this to this, so this is the dihedral angle, and this here is the rotation of the dihedral angle, right? So in this, so how it will be shown, or like rather how this phi and psi will be here for the rotation points. For this dihedral angle in a peptide are defined by three bond vectors connecting four consecutive main chain atoms. So this phi involves, this phi will involve, okay, I'll write it down here. This phi will involve carbon to nitrogen to carbon alpha to carbon. So this is right here, and we'll see carbon to nitrogen, alpha, and to carbon, right? Okay, so for better understanding, I will just take this picture here. So actually just connect this. Alright, I will remove this from here. Just draw it again. I will just remove everything and just I will draw a new picture. I hope that will help us. Alright. So that's oxygen, right? So this is nitrogen, then that's hydrogen, then again it will join with carbon alpha. So then again carbon alpha, then this will be go down, then nitrogen, the hydrogen will go up, then carbon alpha, and then again the same pattern goes on. So this will be like this, and here will be the amino terminus, and here will be the carboxylic terminus, right? So the right to left. So this phi for the dihedral angle, so this rotates on nitrogen to carbon alpha. So this from here, nitrogen to carbon alpha, then carbon alpha to carbon, right? And then carbon to nitrogen. So this is a rotating plane. I will draw it here. That's this, right? This and this. So it will rotate like this. Sorry, it's so complicated, but like just imagine this will be rotated like this. For the phi and the psi, so this was for the phi, and the psi involves, so the psi involves nitrogen to carbon alpha to carbon to nitrogen. So this is how just they describe the rotation along the plane or the bond vectors. All right, and this plot, so there is a plot that is developed by Ramchandran plot, also it's known as. So this plot was introduced by actually G and Ramchandran. And there is a plot that's known as a Ramchandran plot as well. So he described in more details how this rotations goes on. This was just an overview of the protein structure, but we'll see now the protein's secondary structure, right? But this is how this interactions involves, like which interaction, van der Waals interaction, weak interaction, hydrophobic effect that we studied, and this peptide bonds as well, or the covalent bond, right?